Hey, it's Mr. Pletch here. This is a assignment tutorial for Module 4, Viruses and Monera. This assignment is pretty straightforward. Parts 1 and 3 are simulations or click and learn, so all the information is kind of provided as you walk through it. Part 2 is a little more challenging, and I've added kind of a better option to go through that. But it's once again one of those things where it's open and it's totally up to you. But let's just jump right in. You guys know how to find the assignments. So Module 4, the last assignment for the midterm. Once again, 3 parts, 35 marks. Part 1 is a click and learn. And you watched a video for the Ecology Module 3 from the Howard Hughes Medical Institute. This is a great website. They have tons of amazing stuff. I've never personally done this click and learn, but they're organized very, very well. So the idea is you go to the website and you just walk through it, and as you walk through it, you're going to find these answers for part 1. So there's nothing surprising there. When I clicked this link when I got it in docx, it did not bring me there, but you can once again copy paste and search it, or just type West Nile virus click and learn, and you'll find this website. Now, this website won't tell you anything. So to launch the kind of interactive click and learn, you're going to click this over here. And it will open in a separate window. So I believe I already had it open. So you're going to get a separate window and you kind of click through it and learn as you go. So it tells you kind of what's the point of this. There's no questions associated with that. But then the next slide is why study the West Nile virus. When I bring this up, that's one sentence. So please put this in your own words. It's just a couple sentences they say. So I want you to summarize it, one sentence in your own words. What's the point of studying this virus? Now, question 2, compare the prevalence of West Nile virus infections when the virus was first detected to when it peaked. So once again, we're going to kind of walk through it and we're going to get lots of information. This is definitely one of their older click and learns. It seems more like a PowerPoint. There's videos embedded, so if you click those, they'll pop up and help you. How it spread to the peak. So it's all in here. I don't think anything stands out that you need my help with. So just walk through 10 marks, 8 questions. No surprises there. Now, part 2 is all about the lytic cycle of viruses. So as you learned, viruses have 2 life cycles, the lytic cycle or the lysogenic cycle. Now, the lytic cycle is way more simple. They are your viruses that kind of cause things like colds and flus, COVID, so things where they're going to infect you and you're going to have an immune response right away. The lysogenic cycle ends with the lytic cycle, but the lysogenic cycle also has a period of time where the virus goes dormant. So it'll go dormant, you might not know you have it, your body might not recognize it, but then it can kind of kick into gear, start making viruses, and you can get the impacts of it. Or for example, like the flare-up. So herpes, for example, it goes dormant, but sometimes there's flare-ups. Certain things will start the virus into the lytic cycle where it starts multiplying and spreading and causing that infection. Luckily, the lytic cycle is much more simple and just has these 5 steps. So I've updated the assignment. So if you downloaded it before Tuesday, July 14th, definitely look at it again because I did update it with an option 4 and this diagram. But how it basically works is viruses will come into contact with the host cell, and if it binds, it will connect, attach to the surface of the host. After that, this, for example, is a bacteriophage, but it'll basically condense and inject the viral DNA into the host cell, and then the cell will read that DNA and start making pieces of the virus. So we're making tons of pieces, and then number 4, maturation, it's going to assemble the viruses. So that's why there's one of those questions like, are viruses living or dead? And we don't know. Because is assembly really growth? It's one of those weird things. But the virus will be assembled, and over time, they'll become so packed in the host cell that they'll burst the cell open, something called lysis. And then they're going to grab onto another host cell. Once again, they don't know they're grabbing onto it. It's just a chemical reaction of the binding on the cell, and then it starts all over again. So that's the Leydig cycle. And your job is, once again, to demonstrate this in a creative way. And I know you just did that in assignment 3. So kind of the same idea again. Once again, you can make a infographic if you choose, if you liked that idea with that single slide PowerPoint or using those softwares I provided. That's a great way to do it. Very visual, explains everything. You can make a skit again, so you and your friends can walk through where you go infect them and you have some fun. But there's some options given this time. So option 1 is to create a flip book video of the Leydig cycle. So you need to narrate it. You can write it and kind of do a flip book, anything like that. I don't think that's super easy, but if you're a good drawer, that's something you can definitely do. Option 2 is to write a story. So this is where you actually turn the virus and the cell and even the host into characters. And you're going to write a story of the Leydig cycle kind of happening. So what's going on there? If somebody like anime, for example, cells at work, they go through lots of stuff that's like human body. They do bacteria. So I don't know if they do the Leydig cycle, but great videos there. Great series if you want to learn some stuff. So this would be a comic strip. So once again, very visual. You're kind of writing a story. And this one is the one I added. And I said, create an analogy for the Leydig cycle. Now, when I teach Bio 11 in the class, I do an analogy assignment where they can choose to do the Leydig cycle, the lysogenic cycle, or the human immune response, and they have to create an analogy. So this is a shortened version of that, because they have to create an analogy, make a table, and make a visual, and present it in small groups. But basically an analogy is when you compare two things just to make it kind of more clear or obvious. So in science, a lot of the things we learn are quite complex or kind of hard to get our head around. So if we can connect them to things in everyday life or even things you're interested in, it's going to create a stronger connection. It's going to get more people to understand what the process is. So if you want to create an analogy, the idea to get full marks is to create an analogy for each of these 5 steps. And also you need a visual to go along with it. So please don't just Google viral analogy because it's very easy to find. But basically, so the analogy here, so the attachment of the viral life cycle here. So what would that be like in your analogy? And so you're going to just say the analogy would be entering a bank or something. And how is that related? Well, the robber is attempting to get into the bank, so they first arrived at the bank, blah, blah, blah. So it's actually not a lot of writing if you do this, but it's a lot of thinking. A quick example I posted here, and I'll kind of zoom in. This is only 3 things, but we see that first the outlaw will eliminate the town's existing authority, and the host cell's DNA is chopped up. So once again, they're going on their own kind of thing, but the idea is this outlaw has already penetrated the town and is already kind of integrating into the DNA. Now the outlaw demands to be outfitted with new equipment from local townspeople. So this is when the virus or the outlaw is getting them to make weapons or make the pieces for his posse. And eventually when that's big enough, they lyse or leave the town. So they break open the town and they run and they go to the next town. So that would be a very, very simple analogy, but that is just one option you have. I really do like analogies, a lot of thinking, but probably the least amount of work if you can create something like a visual and these steps. Sorry I don't have a better example. All my students typically do the other ones, but that is a good example of it. So that is part 2. Once again, there's so much flexibility. If you have another way to do it, just let me know. And your marking. So creativity and clarity, that's going to be out of 2. And then you have each of the 5 steps and how they're shown. So regardless of what you do, you should realize that each of these steps need to be clearly identified and described. Now, in the analogy, that would be in the how it's related section. So in the virus lytic cycle, it looks like this. In my analogy, this is how it looks. Now, part 3 is the bacterial culture virtual lab. And I wish we were in a regular classroom, because normally I expand my microbiology unit to include 3 different labs. So I teach you how to do the 4-quadrant streak, we do this Kirby-Bauer method, what this is on, and we do an unidentified one where we test some other stuff. But the basic idea here is you're going to walk through an experiment which is actually using something called the Kirby-Bauer method, which is you're going to test how effective certain things are at killing certain types of bacteria. So once again, very straightforward. You're going to read through the background information, and I'm just going to pull it up here. It basically looks like this. Ooh, funny sound. Please go step by step, because I ran into some things I didn't realize were not working. So if you read through, you're going to read the pre-lab, which is over here. Now, once again, pre-lab, you have 4 questions. So you're going to get that all from this side kind of pre-lab thing and be able to answer those questions. Very straightforward. Now your data table is here, and there's one in the lab, but it doesn't really work. And I'll show you what I mean. But your main idea is you're going to inoculate this agar with a certain type of bacteria. So agar or agar is something that's going to stimulate bacterial growth. So it's full of nutrients. It's actually from a seaweed. We use these all the time. You probably used one in Science 8. I use them all the time in there as well. But you want to get a little bit, and you want to place them on each of these circles. That is not the correct format to do that. I don't like this lab because that's not a proper streak, but whatever. So you're going to do that with each of these bacteria. So you're going to have to do everything 3 times. And then you have these 7 little paper discs, which are soaked in a different kind of antibacterial. And so you might click this one, which is our control, just a regular disc. Put it there. You get the idea. You're going to put each of the discs in each of the sections they put bacteria in an incorrect streaking method, but whatever. And then once you have all 7, we're going to incubate. And we're going to incubate for 24 hours. So I put it in, and I'm going to switch on, and it's going to go to 24. 24 hours might be a little bit low. I typically do, if I have an incubator, like at least 48 hours. But whatever. For this one, we're going to pull it out. And we have to measure these things called the zone of inhibition. What the zone of inhibition means is it's an area where the bacteria cannot actually grow. So that chemical is so strong that nothing can grow there. It kills the bacteria. The bigger the circle, the stronger the antibacterial. And that is because out of this disc, it will diffuse. So it'll actually leak out and get less strong and less strong the farther it goes out. And so way over here, it means there's not a lot of bleach required to stop bacterial growth. Versus something like here, it'll leak out all the way, but it still needs a ton of penicillin to actually stop this bacteria. So the bigger means it was stronger. You're going to use the ruler to measure, and it says record them in the table here. This table does not save anything. So you can't actually measure and save it and bring it back up when you do all 3 tests. So please just record your data in the table here. So for example, this is Staphylococcus aureus, so the staph infection. I'll measure 2, which would be 13. So Staphylococcus aureus, number 2 is the antibacterial soap. That would be 13 millimeters. Just so you're aware of the control, if there's no circle around it, that's 0 millimeters. So please fill in that table. You're going to have to do it for every single one of these, so the different 3. And then from there, you just have a couple questions. So question 1 says, describe the effects of the various antibiotic drugs you used. Were they all equally effective? How do you know? So that's where you're going to compare penicillin and amoxicillin on the 3 things. So you're going to tell me which was the best at controlling growth for each of them. So you might say for H. influenzae, penicillin was. For S. aureus, amoxicillin was. That's it. That's all I want to get your 2 marks. And likewise, number 2, I want you to choose one of the chemicals, so not penicillin or amoxicillin, or erythromycin, sorry, that's an antibiotic as well. Which one of these 4 was the best, and defend that. And when you look at the numbers, it's actually pretty obvious which one's the strongest. This should be household bleach, not household beach. And you're just going to say the most effective was because. Other than that, it's really straightforward. I gave another little note at number 5. Can you think of any limitations of this technique? So testing with these little discs. And so just one of the tips I said, is a Petri dish normal conditions? So remember, it's basically unlimited food. So things to think about. What about the time we incubated? So there's a couple things that we could probably do better if we want the best results. But that's it. That's assignment 4. Please let me know if you have any other questions. But yeah, good luck. I think it's pretty straightforward.